Hey, Bio Kids, it's Mrs. Shannon here, and I am going to go over some of the big ideas slash summary slash study guide, whatever you want to think of it as. These are the ideas that I hope that you pulled away from doing your individual learning, I guess, slides here. First of all, we know that gene, alleles, chromosomes, they all go together. So what's going to happen is on the right, I'm going to keep key terms, and on the left, I'm going to kind of explain things. And so here we go. We have our DNA, okay? And my DNA stinks, okay? And if we were to examine it at a macro scale, at a larger scale, eventually this would be winding up into a chromosome. If we take a section of that chromosome, we have what's known as a gene. Okay? So a gene is simply a section of the chromosome that codes for a trait. Now, the variation itself is known as an allele. So let's say that we have I'm going to take one of the easier ones, cleft and no cleft. In your chin, you either have a cleft chin or you have a no cleft chin. Okay? So we have two variations for the allele. The allele is what codes for it. We have either a cleft chin or we have a no cleft chin. Okay? And so these are letters that we use to express what the allele is. Now, the alleles that we use when we say C for cleft, big C for cleft, or little c for no cleft, those are known as the genotype. Now, we get one from each parent. What that means is that since we have a father and a mother, no matter if they live with us or not, but we were born with one, we have two of them. So we could be a CC, big C, big C. Oopsie, I'm drawing. We could be a big C, a big C, or a little c, little c. And that's what's known as a genotype. The genotype just tells us which allele we get from which parent. Now, you notice that we have capitals and lowercase. That's dominant versus recessive. In Mendelian, that's like the easiest, I guess, inheritance pattern that you can think of. The dominant trait, the uppercase trait, is going to be, and that's down here with dominant allele and recessive allele, is going to be expressed as a capital letter. What that means is that there's enough of it expressed that it overshadows anything else. Now, if it's a lowercase one, this is recessive. A recessive allele means that, yes, it's going to be expressed, but it's in such a small amount that it cannot overtake the big one. Okay, cannot overtake the dominant allele. And so we call that recessive. It's not going to show up when it's with the dominant because the dominant just takes over. So far I've gone through gene, allele, I've gone through genotype, I've gone through dominant, I've gone through recessive alleles. Recessive alleles are the lowercase ones. The probability is how likely. Probability is how likely something is going to happen. And we have tools to use to predict how likely something is going to happen with a trait. And so we have two tools. We have the pedigree and we have the Punnett square. So I'm going to get into those in a minute. Lastly, I'm going to erase some of these. Lastly, we have a couple other terms called heterozygous and homozygous. So looking at that gene here, remember we get two from a parent. So I'm going to put those two possibilities up for the cleft chin here. Okay, so remember we have a C. Oops. We have a, maybe, we have a C, C. We have a C, little c, and we have a little c, little c. Now, the thing is that we have ways to describe these genotypes. We have fancy, schmancy words. We have homozygous and heterozygous. Homo means the same, okay? And that was one of our prefixes from earlier. Homozygous means the same. That means it's either all capitals, both capitals, or both lowercase. So we have homozygous, and we have homozygous here, because they're both lowercase in this case. Now, if you have one of each, they're not the same allele, then what we have is heterozygous. So when you're seeing heterozygous, it means we have one big and one little, one dominant, one recessive. Okay? And that helps out when you're doing a Punnett square, for instance. So let's see, what else do we have over there for Mendelian? Oh, okay. So Mendel was this monk who used to observe things in his garden. He used to observe peas growing in his garden, and he used to observe the pea color, the pea style, and the leaf color. So he would say, is it purple? Is it yellow? Is it wrinkly? Is it smooth? And he'd figure things out. And that's what's nice about that is he happened to observe traits that were like the cleft chin, that were easily dominant and recessive. But not everything happens as dominant and recessive. Sometimes we have codominant. So codominant, why am I not writing? Oh, I'm on eraser. Codominant means simply that we are going to show both of them. And so if we have a black cow, oh, don't judge my drawing, guys, okay? Don't judge it. There's my black cow. Woo! And we have a white cow, which you won't be able to see, but you know what? We can still write it. Okay. Now, hypothetically, if one's dominant or the other, you'd have either an all black or an all white cow, depending on which is dominant. In codominance, what's going to happen is that you will see a little of each. So you would see a cow. Let's draw my cow. With maybe black spots on a white background, but you will still see both of them, okay? And so that's the idea with codominance, is that you can still clearly see both of them. On the flip side, we have incomplete dominance. Incomplete dominance means dominance does not quite win. It's a kind of a mixture of the alleles here, okay? And so instead of a black and a white cow, you might have a gray cow. So incomplete dominance means that you have a blend of the phenotypes. It's not quite one way, it's not quite the other. Okay? The last one is sex-linked. Oh, whoops, no, polygenic. I'm going to talk about polygenic in a minute, but you will not... Okay, I want to rephrase that, because some of you guys might choose disorders that are polygenic. But polygenic, poly means many, genic means more gene. So you're going to have many genes involved. You're going to have more than one gene involved. And in fact, most traits that you observe are polygenic. They involve multiple traits. And so that's all that's about. Okay? Homozygous, heterozygous, dominant, recessive, genotype, phenotype. I'm going to talk about Punnett square in just a minute. Yeah. And I think we're ready. So Punnett squares here are tools. They're just a way to organize traits to figure out the likelihood of offspring having a certain trait. So we're going to stick with our cleft chin, and I'm going to call it big C and little c. Now, parents have 2 alleles. So you're going to have a 2 by 2 square, which means that there's 4 boxes in it. So let's say our dad is big C, and our mom is big C, little c. So what we do is we draw a square. It's going to be 2 by 2, 2 rows by 2 columns. Dad is big C by big C, so we put that on the outside of one. Mom is big C by little c, so we put that on the outside of the other. This is how we set it up. And then we just kind of, it's almost like math, I guess. We just kind of fill in the blanks. So big C means that this box would be big C, big C. Big C, big C. Big C, little c. Big C, little c. And by convention, you always put the capitals first. And so this is what the Punnett square would be. So you would have 50% big C, and 50% big C, little c. This would be the genotype percentages, the probability of certain genotypes. Now, if you are looking for the phenotype is the expression. So here we go. Big C equals cleft chin. Big C, little c. Well, big C is dominant. Big C means cleft chin. So it takes over. So in this case, it is 100% cleft chin for the phenotype ratio. Okay? So that's how you kind of work that out. It's really useful for figuring out genotypes and phenotypes. When you look at sex-linked or other kinds of traits, it gets a little bit more confusing. But we shouldn't be focused on that. If you are doing a sex-linked trait for your disorder projects, let me know, because I can help you with trying to figure out how to set up the Punnett square for that. Now, if big C, let's pretend big C was black and little c was white. If this was codominant, we'd have a black and a white for 50% of the organisms, because that's 50% of the boxes. If it was incomplete, we'd have 50% as gray, a somewhat mixture of the two of them. And so you can still use them for incomplete and codominance. Punnett squares are still very helpful tools. So I hope this helps you. And if you still have questions, let me know.